Hello, everyone. My name is In. I'm the product manager from Bio. I'm so glad to see you again in our webinar. Today, we are going to talk about the pharmacokinetic and pharmacodynamic of the ADC drugs. And we will talk about the PK profiles, and we will explore the bioanalysis method about the ADC. And also, in the second part, I will talk about the critical reagents that from Bio for the analysis, especially the anti-payload antibodies and the anti-either type antibodies. First part, I will show you some ADC introduction, and also I will talk about how can we balance the efficacy and the toxicity, and how the evaluation of the PK profiles will be very important. Briefly, at the beginning of the webinar today, I will still talk about the ADC components and the characteristics of the ADC. So as you see here, the ADC are comprising of a tumor antigen-specific antibody. And this monoclonal antibody will bind to the antigen that preferentially expressed on the tumor cells, and also is linked to the cytotoxic drug with the chemical linkers. And after the binding of the antigen to the antibody, the ADC will be internalized and then take the actions. So you see that ADC, they have 3 different components. And I will say that when we performing the ADC development, there are a lot of things that we need to consider, like, for example, for the monoclonal antibodies, we probably need to think about the high affinity of the antibody to the antigen, because the antibody needs to recognize the antigen and then guarantee the specific interaction. And also because the ADC internalization relies on the internalization efficiency of the antibody. So we expect the internalization efficiency of the antibody should be good. And also I know that for the recent ADC, the recent generation of ADC, people prefer to use the humanized antibody. And the humanized antibody, it will reduce the immunogenicity of the drugs. And except the monoclonal antibody, for the linkers, basically we can divide it into the cleavable linker and the non-cleavable linker. And besides the linker, another thing also I want to emphasize is the conjugation method. So the conjugation is between the monoclonal antibody and the linker. So this is to guarantee that the ADC will be, conjugated well with the payload. And the last part is the cytotoxic payload. I know that the payload, based on the targets, the payload can divide it into different classes. And basically right now, the current payloads, they are usually from the natural products or chemical synthesized payload. So you see, ADC is really complex. And then for each part of the ADC, you probably need to, take a lot of things to consider to make sure that it optimized to balance the efficacy and the safety. And for the second slide here, I want to introduce how ADC works. So as you see here, after the binding of the antigen to the antibody, the ADC is internalized into the target cells. And inside the cells, the ADC will be, especially the antibody portion, will be degraded and with the lysosomal degradation. And after that, the drug or the free drug will be released. So there's some possibilities can happen. So the free load can diffuse out of the target cells so one possibility is that it will into the surrounding tumor cells that are not directly bound to the ADC and causing the surrounding tumor cells to be death. And another possibility is that the free preload will get into the on-target healthy cells and resulting in the off-target toxicities. So if the preload get into the tumor cells, it caused the bystander effect, meaning that even though the target antigen expression is not, is not directly bind to the ADC, will still causing the tumor cells to death. So the bystander effect is really important to increase the efficacy. And since today's talk, I will talk a lot about the payload antibody. So I want to spend some time to talk about the class or the diversity of the cytotoxic payload right now in the ADC drugs. Basically, let's take a look at the classic payloads in the approval ADC drugs. They can be divided into main categories. So the first category is the microtubule inhibitors. So this one, they are commonly seen in the second generation of ADC. And another category is the DNA inhibitors. So for the DNA inhibitors, we can further divide it into the DNA damage agent and the topoisomerase inhibitors. And then in this class, this top one inhibitors, there's another famous cytotoxic payload that people may be familiar with it. It's called DXD. DXD is come from the approval drug, the TDXD. This molecule is pretty have a very good behavior because this payload has better bioactivity and also the solubility is good. So which resulting this payload can have a high permeability to across the cell membrane and then to increase the bystander effect that I mentioned before. And in addition to this good behavior, this small molecule has a short half-life. So making that is not over permeable to the cells, causing the extra toxicity to the healthy cells. So except this classic payload, let's take a look at the payloads that used in the current clinical trials. So which means that they some of them, they may be have a very good performance in the future ADC. The main reason that people keep developing to have the new payload is because in the recent ADC drugs, they find drug resistance there. So to avoid the drug resistance, the experiment or to find new payloads are always going on. So to summarize, here I list the characteristics of the ideal payloads that people may expect to see. So for example, the adequately high cytotoxicity and the high stability to avoid the toxicity, and also the increased bystander killing effect and then the proper water solubilities. So here, in addition to this, I want to introduce one more toxicity. Like this type of toxicity is not that familiar to everyone. This one called the on-target off-tumor toxicity. Means that the target therapies, even the target therapies has been successfully targeting the intended receptor, but it will still show the toxicity in the non-tumor cells or healthy cells. Actually, this is not because that the off-target effects actually is on-target effects. It's mainly because the target or the receptor is also expressed in the normal tissues and then resulting less unintended damage to the cells. This is pretty common in the solid tumors because in the solid tumors, the antigen expression is not always that clean. It's meaning that even with the healthy cells, the antigen may have a low expression there. And people always say that when we increase the bystander effect, the bystander killing effect, we can improve the efficacy to the tumor cells. But because this type of the toxicity, we still need to be careful. Because as I showed here, the potency increase, the bystander effect the potency increase, the bystander increase, but still the toxicity, especially this type of toxicity will still increase. So that's the reason when the ADC is, when people are developing the ADC, they always talk about how can we balance the efficacy and the toxicity. I think this is the main reason because there are a lot of things that you are not expecting to see, but they are there. So since we talk about the toxicity and the safety, we all know that the preclinical research to solve this issue, especially for the new drug development, they are very important. And during this preclinical research, it always includes the pharmacokinetics and the pharmacodynamics, and also includes the in vitro stability test, blood concentration time curve analysis. And very important is that when we have the PK profiles and then the PD profiles, we can always build the PK and the PD model to have a comprehensive study about the dynamic process of the drug in vivo. And then from there, we can understand what is the relationship between the drug efficacy and the effective duration. And then after that, after all the analysis and understanding, we can provide the certain guidance to the drug to make sure that it can be administered to the patient, and also to determine the dose frequency and the dosage. So as some other biomolecular, ADC is the same. ADC is even more complex, and there are large differences that you can choose for the ADC. So with the ADC, I will say that the challenge for the PK profiles characterization is always complex and challengeable than all the other antibody-based drugs. And so if someone wondering that why the PK analysis of the ADC is so difficult and unchallengeable, in my personal opinion, I will say that the resulting heterogeneous mixture of the ADC is the reason. And this is because that after the conjugation of the payload to the antibody structure, it will affect the DAR ratio of the ADC. So what is the DAR? The DAR is the drug to antibody ratio. And as I showed here, the payload can be conjugated to the antibody, and also the conjugation location can be different. This is the different conjugation method. And then you can see that the resulting DAR ratio is different for each ADC. And since the DAR ratio changes a lot, it really affects the or resulting the heterogeneous mixture of ADC. And another thing about the ADC is that even at the very beginning, after the conjugation, the DAR value fixed. But then when the ADC drugs enter into the blood circulation, after enzymatic payload metabolism, you will find that the DAR ratio is always changing. So for example here, this is the intact ADC. And when the ADC enters into the circulation, you can find that the payload become too deconjugated from the antibody. So you may have the partial deconjugation, you may have the complete deconjugation. You can also find the antibody is degraded. You can always find the payload is catabolized. So all this, they makes the DAR ratio or the DAR values for the ADC keeps changing. And all this, they are related to the purity, potency, and the safety of ADC. So I think this is another one main reason making the PK analysis of ADC complex. Moreover, I will mention another thing. So I'm not sure people knows that, but I will say the drug itself is unevenly distributed in the tumor tissues. So here's one example. This is the tumor site, especially I think this is the tumor microenvironment. In the microenvironment of the tumors, it's usually filled with the abnormal blood vessels and different groups of cells, and also have the secretive factors and extracellular matrix proteins. So all this, they can become certain obstacles for the drugs to penetrate into the tumor site. So the penetration of the drug is limited by all these physical and chemical properties. So the drug is unevenly distributed in the tumor tissues. Then when we combine this to the heterogeneous nature of the ADC drugs, then things get even complicated. The heterogeneity is becoming like a add-on. So it's synergistic heterogeneity. And I have a result from the literature to showing this. So this is a fluorescent labeled ADC. So when you see the red here, this is the labeled ADC, and the green part here is the tumor site. So you can see that in the merged image here, you can see that the ADC drug is unevenly delivered to the tumor site. And definitely this will affect efficacy. And also it can raise the potential safety risk to less clinical use of ADC. So all in all, I want everyone to notice that there is a synergistic heterogeneity of the ADC, and this is really will be challenging to the ADC bioanalysis. We need to link the efficacy and the safety together to better understand what all the ADC components they are doing, like what they do and how they do. So I talk a lot about the complex of the ADC PK analysis, and hopefully everyone already realize it. And from here, I will have a summary about the analyzed in the ADC mixture that we need to care about, especially when the ADC, after the in vivo biotransformation, how many analyzed will be and how can we to analyze it. So this is a very a summary table about the different analytes that you can find in the ADC mixture. The first one is the total antibody. So as you see here, total antibody is a population that contains the naked antibody and the conjugated antibody. So which means that no matter the payload is conjugated or not, we will analyze all the antibody portion. And the second analyze is the total ADC. Total ADC is this part. So as long as one payload is conjugated to the antibody, we will analyze it. So it means that no matter the conjugated antibody or antibody conjugated drug, we will group them together to the total ADC type. And the third analyte is the unconjugated payload, which is the free payload. So it means that this drug has been completely detached from the antibody portion. And then we will group them together, especially some of the metabolites that has been degraded from the drug. So we will group them together as the third analyte. So for different analyte, we will use different platforms to analyze it. So for example, we have the LBA. LBA is the ligand binding assay. We will use this assay to characterize the antibody portion. And then for the free preload group, we will use the mass spec to characterize it. And above this two, we even have another one. So this is the hybrid assay, based on combine the advantage of LBA and the mass spec. So after we combine them, we can even increase the sensitivity, and also we can avoid the interfering factors that especially found in the clinical samples to increase the specificity and also the sensitivity. So for PK analysis, we know that is important. And then even after we know the type of each analyte, but what would you back to see? So what the result will look like and what you can get from it. Here I showed a very brief example to showing this. So if you take a look at the left panel here, this is a very complete PK profiles for different analytes. So the first is the antibody conjugated drug and then the total antibody. So they represent each portion and they look similar because they have the payload that may be fully conjugated to the antibody. So they look similar even, and then the concentration, the degradation rate is pretty similar. And for the conjugated antibody, for this part, because the payload is gradually released from here, so the conjugated antibody is also gradually decreased. And the third is the free payload. For the free payload, because we know the half-life for this type of the molecular, the half-life is always short. So you see the concentration gradually or quickly decreased. So this is a complete description about each analyte. And for the right panel here, I further separate them based on to see whether the conjugation method can impact it or how stable the linker can also affect the PK fibers. So let's take a look at the top one here. You see that we can use the PK profiles to compare the naked antibody and the total ADC. You see that because the conjugation happens, so the PK profiles between these two populations will also change. And then from there, you can tell whether your conjugation is good or not. And the last here is the about the linker. So you see that for the unstable linker, you see the concentration decreased a lot comparing to the stable linker. This type of the PK profiles can help you to optimize the linker, to assess the stability of the linker. And then from there, you can further optimize the ADC design based on this data. So all in all, this will gives you like good suggestions or mimic the PK profiles that you expect to see in the vivo. So from here, I will focus on the specific analytic method. So the method that I will talk here is the ligand binding assay for the total antibody assessment and the conjugated antibody assessment. The format here is the typical ELISA format for each part of the assessment. So as you see for the total antibody, actually for this assay, we care about the antibody portion of the ADC. So as you see, no matter what the capture reagent we can use to capture the antibody, we always try to capture the antibody. So no matter it's naked antibody or conjugated antibody, we care about the antibody portion. But for the other class, the conjugated antibody, because here we care about the conjugated payload. So the capture reagent for this type of the LBA assay is the anti-payload antibody. So the green one here. So using the anti-payload antibody, we will capture the conjugated payload from the antibody. So to further clear, making sure like everyone know about this, here I made another table to give you details about what is the capture reagent and about the detection reagent. So total antibody, the capture reagent can be the anti-ideal type antibody, can be the target antigen, can be the general reagent. But I will say the anti-ideal type antibody is pretty specific and also is pretty high sensitivity. So it will capture the antibody portion, very specific. And for the conjugated antibody, the capture reagent is always the anti-payload antibody because, as I said, we care about the payload portion. But the detection reagent is similar to the capture reagent here. You can choose different formats, but I will still say the anti-ideal type antibody is always the, I would say is always very specific, yeah, comparing to the others. And then the last part in the last portion in the first part of today's webinar is the DAR ratio change, what I mentioned before. So DAR ratio is the therapeutic index of ADC. So when you understand, after you understand the DAR ratio change, you can always make a prediction about this ADC's efficacy and the safety. So generally for this type of the assessment, we use the affinity mass spec to perform the analysis. So generally, we will isolate and enrich the ADC molecules from the complex biological matrix. And so to say is that we use different capture reagent to capture the ADC from biological samples. And usually, as far as I know, commonly right now, this assay is in the form of the magnetic beads. And after the enrichment and isolation, the different alternative analytes, they can be released from the ADC. So for example, if we care about the antibody portion, we can perform the hydrolysis to degrade the proteins, and then we can use the mass spec analyze to analyze it. And if we care about the payload, then we can use the drug assays to release a free payload and then perform the mass spec to capture it. So this is pretty like, I think it's basically the same as what I mentioned before, the hybrid assay. So combining the LBA together with the mass spec. So with both of the advantage, you can always increase the sensitivity and the specificity. Here is an example about how would you expect to see about the results. what the change of the DAR ratio you can see when you perform this type of the assay. The example here is the ADC drug called the TDM1. So in the TDM1, the DM1 is conjugated while the lysine residues. So after the conjugation, the different DAR value species, the DAR value can be the value from the 0 to 8. So at the 0 hour, this is a typical distribution of the DAR. You see that you have different species here from 0 to 8, right? And then after 24 hours, you can clearly see that there's a drug deconjugation happens. And then resulting the higher DAR species, especially the DAR 6, 7, they disappeared and they kind of like transform into the low DAR species. So you see that the DAR 1 to DAR 3, they're enriched. And at the 96 hours, you see that the complete disappearance of the high DAR species, like the DAR 6 and DAR 7, they disappeared. And you see a very significant increase from the DAR 1 to DAR 3. And all these three, they have a relative abundance. And we also, and then in the literature, there's also another group, the buffer control group. And if you compare this one to the 0 hours, they look similar, right? So this is how we expect to see for the analysis about the DAR ratio change. And this can help us to understand the in vitro stability of the ADC drugs. After I talk a lot about the PK analysis of ADC, hopefully you understand the importance of it and then what analyze that we need to analyze and what type of the analysis method we can use. And then for the second part, I will talk about the critical reagent from Bio, like how can we, what type of the reagent that we can offer to you, including the anti-payload antibody and anti-ideal type antibody. So first is the anti-payload antibodies. Bio provides different anti-payload antibodies corresponding to the different payloads that people commonly use in the ADC design. So the first is the popular DXd molecular called the DXd payload. For this one, we have two different clone number, the clone number for the DXd monoclonal antibody. And also we have the HRP, the labeled anti-DXd antibody. So with the HRP conjugated antibody, we can facilitate the detection and eliminate the need for the use of the secondary antibody. And also with the DXd payload antibody, we also have the recombinant monoclonal antibody. This is to make sure that if you want to perform the large quantities of PK analysis, this type of the antibody will have a very good consistent and and then making sure that your data is reproducible. So to control the batch to batch consistent. And except the DXd payload antibody, we have some other payload antibody, like the one that I mentioned, the microtubule inhibitors, the anti-MMAE and MMA payload, and anti-eribulin payload, anti-SN38 payload. So this payload is currently, they are all in stock. If you want to find more, please go to our website. And since I know that the researchers are developing to find more payload. So our product is always increasing. So more is coming. You can kind of like expect it. Besides the products, we also perform different validation experiments to making sure that what we provided to our customers is with good quality. So here is the validation data from a functional ELISA experiment. So with all our anti-payload antibody, we verified the binding affinity for all of them to the toxin. So for example, this is the functional ELISA between the anti-DXd monoclonal antibody to the DXd toxin. As you see here, the binding affinity of them, they are pretty good. And also they have a very good sensitivities. Except that in addition, we also guarantee that our anti-payload antibody will have a very good purity. So the purity is more than 90%. It's validated with the SDS phage and also with the HPLC. And moreover, except the binding experiment with the true toxin, we also performed our validation experiment with the clinical ADC. So for example, here with our anti-DXd monoclonal antibody, we performed the binding experiment with the TDXd. So TDXd is the approval ADC already in the market. And from our binding experiment, we confirmed that our anti-DXd antibody can successfully bind to this drug, so which is very exciting. And the same as the MMAE and the MMAF monoclonal antibody. These two drugs we used here, I think they are either from the approval drug or the clinical drug. And our antibody can perfectly bind into them and then, have a pretty good sensitivity and high affinity. And moreover, we also perform some other comparison with our competitors. So this is an experiment that we performed between our product to our competitor's product. So these two lines here, the pink one and the purple one, they are two anti-DXd monoclonal antibody from our competitors. And then we did the parallel comparison between their antibody and our antibody. As you see here, the results showing that the antibody activity of ours is 4 to 5 times that of our competitors. This makes us so exciting, and then we are proud to say that right now our products look so excellent and beautiful. Besides the anti-payload antibody, Kussobio can also provide the customized service to produce the anti-ideal type antibody for more ADC PK analysis. So one example here is the monoclonal anti-ideal type antibody that we can produce. This type of the monoclonal antibody is usually used to detect and quantify the drug levels in the animal or human serums. So we know that before the clinical research, we need to assess the blood drug concentration based on the time curve. And this will refer to a dynamic process of the blood drug concentration change within the time. So with this capture ELISA based on the anti-ideal type antibodies, we can perform this type of the PK analysis to analyze the dynamic change of the drug concentration and quantify it. And, this is kind of very required based on the FDA requirement. So if you want to have this, if you want to perform this type of the PK analysis, Kussobio can give you a customized service to produce this type of antibody. And moreover, with the anti-ideal type antibody, you can perform another type of the PK analysis called the immunogenicity assay. This assay is kind of different from the previous one I mentioned, because the previous one, you use the monoclonal antibody, but here, for the immunogenicity assay, you probably need the polyclonal antibody. Because here, this is to get rid of the risk that of the existence of anti-drug antibodies. So you may wondering what are they? So the anti-drug antibodies sometimes will be produced after the administration of the biological drugs, especially the antibody-based drugs. And their presence can bind to the active site of the drugs and then inhibit the activity of the drug. So you can use the anti-ideal type polyclonal antibody to perform this bridge ELISA assay. And in this type of the assay, the anti-ideal type antibody can be used as a positive control, and also it can be used as a reference standards to assess the immunogenicity. So think about that. The anti-EDO type antibody, they are highly specific for the ADA. So it can mimic the real situation of the ADA in your blood samples. So using this one as a positive control, you can kind of like predict that what is the ADA in the blood samples about your drugs. So to be summary, for the anti-EDO type anti production, you have two options. Really based on like what is the application of your product. You can choose to customize a mouse monoclonal antibody or a rabbit polyclonal antibody. So the difference is that for the monoclonal antibody is really high specific and the sensitivity. So you can perform the PK analysis to quantify the drug concentration from the blood sample. And then for the polyclonal antibody, you can perform the immunogenicity evaluation because this one will mimic the ADA situation in the real samples, and then to tell you that how good is your sample and how many ADA is in your sample. So Kusal Bio has years of experience in the antibody development. So we can customize the anti-EDO type antibody and then provide these solutions for our customers for the PK and the immunogenicity analysis. So overall, this is the end of our talk today. And please give me 7 minutes to tell the story from our company. So our company is Kusal Bio. This comes from a promise from our company to our customers. So Kusal Bio means customer satisfied biology. And right now we have a different lines of product that we can offer to our customers. So like we have the ELISA kits, we have proteins, antibodies, and the molecular biology product, and also the small molecular antigen. And our company has been exported to more than 80 countries, like Europe, America, Japan, and Germany. And also we have a very good cooperation with famous companies such as Pfizer, Sanofi, Bayer, and Moderna, and Zinco, Johnson & Johnson. So you see that we have a very good relationship with these famous companies. Then this is the end of the talk today. Thank you so much. And if you want to communicate with us further, please check us in the linking. And this is the scan code. You can just directly scan it and stay connected with us. Thank you today. And I will take the questions.